Today I would like to talk to you about how to calculate the diversity of a particular field site. The simplest measure that you can use is richness, which is just the number of taxa. And for reasons that I'll go through in the next slide, that is, while it's simple, it's not particularly informative. And so the way we're going to suggest that you do it is to use the Shannon-Weaver, often just called the Shannon Index. Richness, as I said, is the simplest member, and it's simply the number of taxa that you find. And it could be species richness, it could be family richness, it could be order richness. Whatever level of taxa you are working at, then that can be the richness. It's very simple to calculate. Here's the data from the spring 2014 Streamlab that we collected. And if we just count up the number of pieces of pies, we see that the richness is 16. There's a problem with this, though. Take a look at the size of those pieces of pie. Some of the taxa that we collected are available at fairly high levels. For example, almost a quarter of the sample is Ephemeralidae, whereas other taxa are there at very small levels. There was 1% of the taxa were Peltoperlidae. 1% was Brachiocentrids. By using richness, we weight each of these taxa equally, and it can lead to a misleading picture of what your stream actually looks like. For that reason, we suggest that you use something like the Shannon Index. The Shannon Index, which is symbolized by H, combines richness with how many individuals are in each taxa. So here's again the stream 2014 stream data in a slightly different format. The numbers represent the number of individuals caught out of all the samples. And we can calculate the Shannon Index, and it will take into account both the fact that we have 16 different taxa in there and the fact that we have 17 Setipalpian or Perlidae and 94 Ephemeralidae, but only 1 Batistidae. The formula for the Shannon Index is very simple. It's just symbolized by H, and it's the negative sum of the proportion that each taxon is in the population multiplied by the natural log of that proportion. And I think the easiest way to explain this is to show you using some data. So I'm going to switch to Excel. Here's my Excel spreadsheet. Again, it's the same data that you saw before. The first thing I'm going to do is I'm going to figure out the total number of critters that we caught. So I'm going to use equal sign sum and just sum up the total number sampled, like that. And we found 387 insects in total. I'm going to make that bold just so that I remember that. And I'm going to write the word total over here so I remember that. Now I'm going to set up a column, and I'm going to calculate the PI for each taxa, or the proportion that each taxon is of the total. That's very simple. Equal sign, the number of Batistidae that I caught, divided by the total number of insects. And I get a value. And I can just use my drag and fill option to fill that in for each taxa. Oops, there's a problem here. I've got to divide by 0. And then I remember that what Excel is doing is that when it tries to calculate this, it takes this number, but what value would be in the cell below that. So I need to go back up into this cell and say, I always want you to use my total. So I'm going to put in my $ characters. Now when I drag and fill, boom, I get all my PI numbers. Now what I can do is go to this cell, and I'm just going to use those PI numbers to calculate the PI times the natural log of the PI. So PI natural log, which in Excel symbol is ln, PI, click in this cell, type an equal sign to tell Excel I want you to do a calculation. I want you to take the PI, I want you to multiply that by the natural log of that number. Whoops, somehow I need a bracket in there, end bracket, there we go. And I can now drag and fill. And then to calculate h, I need to sum that, so equals sum. I've got a negative number here, and I remember that my Shannon index is the negative of this number. So I'm going to take that, and I'm just going to take the absolute value of it. Excel language for absolute value is ABS. And there we go. My Shannon index for the stream is 2.22704729. Now that's way too many decimal points. I'm going to keep all my decimal points for the calculation, but I'm going to round down to something more reasonable, 2.3. And that's my Shannon index. Now a single value for the Shannon index really doesn't tell you much. It is a good starting point, but where it can be really helpful is if you could compare Shannon index from one time to another time point, or from one field site to another field site. We're not going to ask you to do that, but we are asking you, if you're doing the forest project, to calculate the Shannon index and report that to your landowner. And that's how you would do it.